What we'll talk about today is a newly developed test for the presence of E. coli in the beef cattle chain. TG, would you outline the seriousness of this condition and the considerable research that's going into it now?
Okay, Eric, E. coli 0157H7 is a type of E. coli that is present in the gut of cattle. Therefore, it gets shed in the feces of cattle. But if that organism gets into human food chain, it can cause serious infections in humans.
It has drawn so much attention in recent years, and in fact, considerable research being invested in it. The work we'll talk of today is supported by a USDA grant on Shiga toxin-producing E. coli 0157H.
Yes, K-State and University of Nebraska are the two lead institutions in a multi-institutional and research institute grant given by USDA. This is a $25 million project. So the goal of this grant is to understand more about these 7 different types of E. coli, and all are present in cattle and shed in cattle feces. Our team is part of Objective 1, which is to develop new diagnostic methods. And that's where we have developed this procedure that could detect as well as quantify E. coli 0157H7 in cattle feces. And that quantification is important because typically most cattle shed E. coli 0157 at low concentrations and intermittently. But there is a subset of cattle population, small number, that shed E. coli 0157 at very high concentrations. And those cattle, there is enough research done to show those are the primary source of contamination. They not only spread the bacteria to other animals, but in the packing plant, if the carcass gets contaminated, oftentimes it is because we have 1 or 2 animals shedding at high level. So the method we developed is to quantify. That way we can identify the cattle that are shedding at high concentrations.
This is a new testing method for Shiga toxin-producing E. coli 0157H7. And Jianfa, can you explain the approach here for us?
Sure. This particular approach we call real-time PCR means that you can detect the concentration, as TG mentioned earlier, as opposed to the traditional endpoint PCR that you have a fluorescent probe involved. So each cycle of amplification, you are monitoring the amplification through a digital camera on top of the reaction plates. So once you see a signal, then it's captured by the camera, so you know when at which particular cycle the signal is captured. Then you can reference back to bacterial concentration. But the novelty for our procedure is to have 4 different molecular targets in one reaction. What we have here is to detect 0157 target along with 3 major virulence genes.
And Lance, for further comparison purposes, when this test is employed in time savings, because time is critical here, this is quite an advance over the previous time span of previous tests, right?
That's right. Traditionally, we've had to culture these out where we can directly take a fecal suspension, run a real-time PCR on that, get results within 2 hours. It really leads to potential rapid automation of this process.
So the process has been tested out here at Kansas State. Do either of you see it being implemented as a routine in the industry here very soon?
I think it is possible. Once, because we will have this procedure published, and there may be companies that may take that concept and make it an automated procedure. That way, all they have to do is inject the sample into a machine, and the machine will give the results as whether it's positive or negative.
On site, then, as opposed to having to deliver it to a laboratory.
Right. And might also tell you at what concentration the bacteria are in the sample.
Well, congratulations to the three of you on this development, and looking forward to hearing more about it, as it may well start to enter the routine of testing for E. coli 015787 and the other Shiga toxin producing E. colis out there.